This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1079, recorded on January 12, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, everybody, and a Happy New Year again. Looking out my window, it looks kind of not great. This morning it looked terrific. It was absolutely crystalline. And now the clouds are moving in, and we're told that by tomorrow we should start building an ark.
In some parts of the country, it's snowing, Dixon.
Yeah, and in that case, get my mushers out.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here is one of the places where it's snowing. I came home from work early so that I would not have to mush home afterward, because at first they were talking about 7 inches of snow by 6 p.m. and then it's dropped off and it started later, and it's above freezing here. So it's 34 Fahrenheit, about 2 Celsius. And it's supposed to turn to rain, but there's so much of this that it could be the flood that you guys are going to get in New Jersey.
Wonderful. Right. Thank you very much. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. Good to be back. It's been a while.
Where have you been?
Around.
Okay.
Christmas was on the West Coast. We spent time in Bend, Oregon, at my wife's vacation house in Sun River, and spent time in Pasadena at my stepmother-in-law's 90th birthday, attended by 45 of her family. It was a surprise.
Wow.
A third of those, 15 of them, were my sort of, offspring and offspring. Okay.
Cool.
It was the first time we've been together in 6 years, 5 years, something like that. So that was all good.
Wonderful.
Does she live in a arts and crafts house?
In Pasadena?
You
mean a craftsman house?
reference.
There's a ton of them in Pasadena.
No.
The Green and Green brothers,
Nope.
back at the heyday of arts and crafts, built homes in Pasadena that are now international tourist destinations.
No, she lives in a nice flat.
Okay.
Right off Orange Grove Boulevard, where they assemble for the Rose Parade. So
And that's where the ultimate game in college football was held this year, right?
Yep.
Well, the
So
penultimate
I
game.
do need to report that it is clear skies and windy. And tonight they're talking about 35 degrees, but then 1, 2, 3, 4 consecutive nights of sub-freezing temperatures, which is a big deal here in Austin, Texas, even down to 15, but with a bit of a thaw during the day. So Jack Frost is going to come through and kill all the plants that we try to maintain foolishly. That's good.
And all the citrus also.
Yeah, there's not a lot of citrus around here.
There is in Texas, though, right?
Yes, elsewhere, I think.
Whereabouts?
Dixon, well, let me introduce everybody. Hang on. then you can talk.
I haven't talked to Rich in a long time.
I'm done.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And it's pretty similar here, 45 Fahrenheit, 7°C. A few clouds up high in the sky. Very pleasant right now, but yeah, as has been said, après TWiV le déluge. So
Indeed.
it's supposed to rain cats and dogs tonight and flood the rivers and all that sort of stuff.
And
a gully
from
washer and a trash mover, as some of my kinfolk would call it.
And from Madison, New Jersey, Brianne Barker.
Hi, great to see everyone. My weather is exactly the same as Alan's, 45 Fahrenheit, 7 Celsius. And we are in between floods here in New Jersey.
Same here.
So right now it's gray and dry, but that won't last long.
Bound Brook won't be for long. Yeah.
Or the Raritan River.
Have you seen the Passaic?
And the Passaic.
If you enjoy our work here at TWiV and Microbe TV, all the other podcasts, please consider supporting us. We need your help to continue. Microbe.tv/contribute. Kathy, what's going on with ASV?
Well, it's fast approaching the time when you should be getting your abstract ready. They are due February 1st. And if you're a student or a postdoc or teacher of undergraduate virology and you want to apply for a travel award, you can also start working on that application. They are due several days later. But students and postdocs need to be ASV members. And so it's not very much, but you can save yourself some anguish if you go ahead and apply for your membership right now before the rush. And so you can visit asv.org for links to the membership page and the meeting page. The meeting, as we've discussed before, is in Columbus, Ohio, June 24th to 28th at the Greater Columbus Convention Center.
Guess who has not yet renewed his membership?
What happened to that sticky note last week?
So it was on my laptop for a few days, and then it got broken off in the backpack, and it disappeared. So I need a new one. But you know, folks, we have Microbe TV sticky notes.
I know you said you were
That
going to send
apparently
us
fall
some.
off your laptop and then you lose track
Well,
of what
good
you're
are they?
doing.
I think all sticky notes fall off.
Yeah,
but,
they.
I tried to. So after
You
the
don't
last
have any
TWiV,
kind of. You don't have alternate ways of keeping track of things you need to do.
I have many ways, and I don't pay attention
You
to
don't
any
pay
of
attention
them.
to any of them.
That's
Okay,
the
yeah.
problem.
Kathy, you texted me about it, and I did do mine. Did you
Good.
text Vincent also?
She did. So I
Yeah.
tried to get on. I went to the site and I said, "There's no link here for membership." So I texted Kathy. She gave me the answer, and then that was I just
Failed.
was doing something else, and I never got back to it. But maybe this weekend I can do it because I really would like to do that.
Okay, I'm making myself a sticky note.
always a member.
You have
mean,
a life
maybe I-
membership?
I do.
Maybe I
Yeah.
should do that.
It's simpler. I wish I
Yeah.
had done it a long time ago, only my grants wouldn't pay for it. The department wouldn't pay for it. Okay.
Yeah, I'll pay for it myself. That's okay. But I have certainly wasted more money than that on
Yeah.
silly things. So I should just do that. And then I don't have to do this every year. All right. Don't forget there is a research assistant position in Amy Rosenfeld's lab at the Center for Biologics Evaluation and Review at the FDA to study enterovirus pathogenesis, the role of cross-reactive antibodies, a little bit of pathogenesis, all BSL-2 work. And there will be a link in the show notes to a PDF. If you don't know what show notes are, you just go to microbe.tv. You could do /TWiV. You get the TWiV page, and then you'll see the episode. And you click on it, and there are links to things we talk about, like ASV and microbe.tv/contribute, et cetera, all the papers. All right, so look for that. And also, don't forget Dixon's book, The New City. You can buy it at despommier.com.
Is it?
Dixon was-
So I just ordered it from Amazon yesterday. It's supposed to arrive today. Looking forward to it, but would it have been better to buy it on despommier.com?
No,
No,
they
no,
just-
it's a link
Okay.
to Amazon. It's
a link to Amazon. It's
Thank
just
you.
a
Thank
landing
you.
page where you can go to Amazon or where else. There's a picture of Dixon. There's a blurb of Dixon. You know,
Yeah,
the usual.
yeah.
block all that out.
Or as someone once called it, a vanity page, right, Dixon?
Well, that's right. My book
Could be a
agent said
vanity
you got to
page.
have one.
Dixon, you have a new headset today?
No.
Oh, okay. Looks
Is
different.
that a
So-
sticky note item?
A friend of mine, not too far away, said, "I just bought your book. I'm going to send it to you. I want you to sign it. I want you to send it back to me."
Oh, great.
And when I added up the cost of sending it back and forth and the option of maybe getting it lost, I said, "Why don't we meet for lunch and I'll just sign it?"
There you go. Good idea.
Well,
yes,
it depends
I
who
think
they
that
are.
would be much better if I wanted mine signed.
Let's do that.
Works for me.
Come on.
Kathy, you put in this note about Elliot Keefe.
Yes, he passed away. He was a lifetime member of ASV since the founding of ASV in 1981. He's been a member.
See, he
So-
didn't lose his sticky note.
Yeah, we have
didn't.
a link to an obituary on the in memoriam page of ASV. Right now, his first name is misspelled, but we're getting that corrected. And it doesn't really cover any of his science. And so I have a line in to someone who says that, yes, somebody's going to write some scientific tribute. And when we get that, we'll add that to the page. So,
All right, a couple of very brief news items today before we get into our papers. There's a report at WHO. It's actually from December 21st, but not so long ago, that global dengue cases have risen a lot in the past 2 decades. Cases rolled tenfold across 129
it's
countries.
amazing.
Yeah.
Yeah. This is not just improved reporting. This is a real and major
Now, this is
trend.
This is a big deal.
Factors include vector distribution changes, climate change, El Nino weather pattern consequences, and fragile health systems. It's all the perfect storm,
to put
right,
numbers
Dixon?
on this, the tenfold increase was from 500,000 to 5.2 million cases. And is that annually?
I think it's between 2000 and 2019.
Well, right, but you would expect the number of cases to increase over time if you just counted every case once, and the numbers would get bigger, and eventually they'd go.
Yeah, I don't know what
infinite,
that number is.
So, I think that may be annually.
I read this as annually.
Yeah.
Well, we could go to the
incidence and prevalence.
I think all of those reasons come under the heading of "Duh, I told you so."
Oh, so
Right.
basically in 2000 there were 500,000 cases, and then in 2019 there were 5.2 million. So it slowly increased per year.
And 2023 there were over 5 million. Yeah. Okay, so that is annual. That's a lot of people.
Is
That's a
that
lot
coincident with the pandemic?
So
No, as a matter of fact, took a dip.
Pandemic
during the
actually helped. It reduced these numbers a little bit because people were out and about less, but not back to 500,000.
No, maybe when they were released from the pandemic, maybe they over.
Yeah. So I mean, the things causing this, as Rich said, it's environmental degradation and climate change, and the mosquitoes are more widespread because they've been shipped all over the place, and so you got more vector. You've got their vectors are surviving better because of higher rainfall and higher temperatures, and it's all just. This was predicted by people 20 years ago.
Dengue said there are too many people. Let's see if we can do something about this.
Yeah.
Mm-hmm. That's what parasites do, right, Dixon?
Yes, but the other thing I think about, everybody blames airplanes and people travel for this, but the number of hurricanes has gone up tremendously over the last 20 years. And hurricanes are great distributors of mosquitoes.
And I would point to shipping as a bigger factor than airplanes.
Also bigger. That's an airplane, of course, and boats. Well, maybe through the Straits of Hormuz, they're not as readily distributed as they used to be. Bad joke.
Shipping in particular moves the mosquitoes around.
Absolutely. Yeah. Absolutely.
Speaking of outbreaks, the city of Philadelphia that Alan knows well has had a measles outbreak. Cluster of cases, all among unvaccinated residents. I think there was an index case who came from overseas and went to a hospital, right?
Yeah. So I gather that maybe I'll let Brianne take it.
So I was talking with someone about this yesterday, and it seems as though it was an index case who was traveling, who then was treated at CHOP.
Yeah.
Thomas Jefferson.
Oh, okay. But it sounds like there has been spread at CHOP.
Okay.
Speaking to a parent of a patient there, potentially because of kids there being unvaccinated or immunocompromised.
Yeah. So the index case seems to have been an incident. It was, it was announced December 23rd. Oh, December 19th, at a Jefferson Health building. Patient, I guess, came in, and people in the building have been informed. If you were in this building, if you were at 33 South 9th Street, December 19th, between 2 and 5:30 p.m., you've been exposed to measles. And they've now had some subsequent cases pop up, and as Brianne just filled in, some spread apparently at CHOP. which is Children's Hospital of Philadelphia, which is definitely a place that could get that kind of thing going.
Yeah.
I mean, not because it's a bad hospital, but because it's full of kids.
They
And
didn't
it's full of kids who might have serious illnesses, who might be immunocompromised.
Yeah.
They didn't elaborate on unvaccinated, whether this is deliberately unvaccinated or what. Because this is a little tricky, right? Because there's this with, correct me if I'm wrong, but with measles, you have maternal immunity that hangs on for a while, so you don't want to vaccinate right away because it'll compromise the vaccination, but you need to vaccinate as soon as possible after that wanes, which is, I think, at about 6 months. So it's a kind of a crapshoot in between there, right? I mean, you could get, if you're exposed at less than 6 months of age before you've been vaccinated, you're potentially susceptible. I got all that right?
I think all of that is right. And at least from the information I learned through the outbreak itself, both groups were involved.
That is deliberately and not deliberately unvaccinated?
Yes.
Okay.
Yeah, I think there were some who, where the vaccine was losing efficacy. Let's see. Where did I see that? We see cases declining vaccination, failing to adhere to quarantine. Okay, so no, it's declining vaccination and not quarantining. So if you've been in these places, they're saying, and you don't know if you've been vaccinated, you should get vaccinated.
They do have this encouraging statistic that Philadelphia has a generally high vaccination rate. 93% of kids are fully vaccinated against
That's
measles
good.
by age 6. So that's good.
But as you
But
see,
if you're in that 7%,
Yeah.
you've got like a 1 in 20 chance of landing in the hospital if you
Yeah.
catch this, and sequelae can be really bad.
what was the statistic I just read? If so, yeah, 93% by age 6. However, 90% of people with close contact with an infected person will get measles if they are not vaccinated.
Right.
And as
That's it's really contagious. That's an
Yes.
astonishing number.
And then, as Alan said, 1 in 5 will be hospitalized.
1 in 20. Or, oh, no, right. 1 in 5 go to the hospital. 1 in 20 get pneumonia.
Pneumonia, yeah. And 1 in 1,000 get
1,000 get
encephalitis. So none of this is good, folks.
Those are not odds you wanted.
Please vaccinate your children. And so last night on the clinical update, Daniel said to me, Vincent, do you know when the last outbreak of measles was in the U.S.? And I said, it was right in the show notes. I could have read it, but I didn't look at it. And I said, Disneyland. He said, no, Rockland County, New York. And then before that, Disneyland. Yeah. Large number of unvaccinated people in Rockland County. And Disneyland, which is out in California, a lot of people didn't vaccinate their kids. They all took them to Disneyland, and there you go. So don't be silly, folks. Don't be
goofy.
Don't be goofy. Okay. We have an interesting snippet for you today that I have been wanting to do for a while, and I don't know why I didn't do it. I felt kind of not qualified to talk about it. And nothing has changed there. I'm still not qualified to talk about it.
He didn't suddenly get qualified to talk about it.
No, but I think it's interesting, and I'm sure there are people out there who know about mineralization, but it's in scientific reports, and it is entitled "Viruses Participate in the Organomineralization of Travertines" by Slokowitz, Perry, Talyasaki. Tie the bag, or cut the bag, one of those two. Powell, Dzilak, Borkowski, Gradzinski, Kell, and Tucker, who are from, they're from Poland and
University
Italy.
of Warsaw,
Hungary.
Krakow, Budapest.
Bristol,
Università della
UK.
Calabria in Italy.
Yeah.
Pamukkale
Travertine.
University in Turkey.
So this is about travertine.
Which of course,
is,
then we all had to learn.
Yeah.
That
we all
was
had to learn about
travertine.
travertine. You know what travertine is? And it's
Yes.
Dixon, you know what it is? You want to tell us?
Well, no, let Kathy tell us and quote Wikipedia because they're wrong.
Well, I somebody else put in the Wikipedia link, and all I added was that it's like what's at the Yellowstone Minerva Terrace.
Yeah, that's right.
And the most famous feature at the Mammoth Hot Springs is the Minerva Terrace,
That's
which is a series of travertine terraces that were deposited over many years, but due to recent earthquake activity, the spring vent has shifted, rendering the terraces dry. And so the picture I put in is from the recent time when it was dry, and I was very disappointed because it wasn't like I remembered from the 70s.
You're right. And it's shifted. There is actually a small active portion. If you are entering Mammoth Hot Springs, you can see it smoking off into the distance. I used to camp there a lot when I was fishing Yellowstone, and it was amazing to see it. But I had no idea that there were viruses in my footsteps.
Dixon, what is wrong in the Wikipedia post?
It's got an official name, Mammoth Hot Springs. That's its name.
Well,
And
this
it's
is
Mammoth
just
Small Hot, and then parentheses Springs.
no, Dixon. I quoted from a part within the Wikipedia that just says
I'm not blaming you.
No. And so then I inserted those other things. And so we had this little editing Google Doc war. But,
It's the official title of that park is Mammoth Hot Springs.
of that part of the park.
So when I went to look up travertine, I got images of what the stone looks like,
Yes.
in your bathroom or whatever.
Yes.
And I've been to Mammoth Hot Springs. I know about this kind of stuff, though I didn't know much about travertines, but I certainly never made the connection to the stone that you see all over the place.
Yeah, the calcium carbonate. It's just another name for calcium carbonate.
It's a limestone. It's
exactly right.
It was used a lot since ancient Rome to tile and build things, and it's structurally strong. And but it forms in places like hot springs, and you can also get travertines forming in caves where you have limestone-rich water dripping through, and it forms cold instead of hot. There's some structural differences, but this is about I think this is mostly about the hot spring.
It is.
And the general idea was that this was a kind of chemical process of mineralization, in whatever of those locations it's happening in. But this paper is sort of indicating that no, in fact, viruses are also involved.
Well,
And bacteria.
Yeah,
Yes.
my guess is that for a long time it's been understood that the way I conceive of it anyway is that at the sort of the growing edge of these things, there's a biofilm,
Exactly.
right? Which is
That's
a
right.
whole
That's right.
mostly prokaryotic ecosystem. All right,
Archaea, lots of archaea.
that's potentially, well, probably involved in the mineralization. In that, a biofilm is a structure.
Right.
So what's in the biofilm?
Yeah, so these travertines are formed by precipitation from these waters, these high temperature waters. And as Rich said, there are all kinds of living things in there: bacteria, fungi, algae, bryophytes, vascular plants, and they all contribute. So it's not just a mineralization; it's also a biological process. Why?
Biomineralization.
Biomineralization.
It's almost like fossilization.
And so it's been suspected that viruses are involved, but no one really looked at it very carefully. And that's what they do in this paper. And they take biofilm samples from various places where travertine forms. It looks like one in Italy and one in probably Hungary, I guess.
Mm-hmm.
One in the UK and Bath.
One in Slovakia.
Okay. And then they look at these samples and they do stainings. And, for example, fluorescent staining, they can see bacteria, they can see cyanobacteria, algae, pollen. You just look at them under the microscope.
Right.
And they said they could see some what look like virus-like particles. And so they do transmission electron microscopy, and they see 80 to 300 nanometer sized particles, and they range from non-mineralized to fully mineralized. So, some of them are completely saturated with minerals because their idea is that these virus capsids attract minerals and nucleate the mineralization. So these virus-like particles are consistent with virus, capsid.
And they're basically catching them in the act because they have them non-mineralized and partially mineralized and fully mineralized, and you see these viral particles, and looks like the viral particles are participating in or
Yeah.
are part of this mineralization process and building the structure of the stone.
Yeah, so they have a variety of photographs so you can see what look like virus-like particles. They also do some sequencing of the nucleic acids from these samples, and as you may guess, they find viral DNAs, particularly DNA viruses with icosahedral capsids, no viruses with an envelope, and the viruses they find seem to infect bacteria and archaea. So, as they say, it's difficult to demonstrate that these are viruses, right? They look like
Very.
viruses,
Well, a little bit.
and just because you find viral DNA doesn't mean that it is part of those, right? So it's a tough thing to do.
But the
But,
temperature.
if
Temperature.
there,
Go ahead.
go ahead. You go ahead.
I was just going to suggest the temperature is very, very hot in these hot springs. People die by slipping into them, and they don't come back out again. They mineralize as well. So to realize that the DNA molecule survives at that temperature, although of course that's what you do when you anneal it. The PCR. I guess. Are you surprised, Vincent, that you could get a good sequence out of that?
I'm sure they're just small bits of sequence, right? Short reads, it's fine, but
We've
it's enough
gotten
to identify
really
them.
good at recovering DNA from things and
Yeah,
sequencing it.
that too.
And DNA is everywhere, right, Vincent?
Yes,
It's
a real problem.
It's a real
It is.
problem. It's a real problem. So, they have virus particles here, and they say in their discussion, there have been other experiments done that show that viruses can bind to mineral phases. They can influence mineral formation. They can affect particle size in calcium carbonates and the formation of various mineralized things. So it's all consistent with it. And so what they think is going on is that what they're seeing are mineralized viral capsids, right? It's kind of like a fossil, right?
Yes. Exactly.
And they say the capsids are mineral nucleation sites. They're negatively charged, right? And they attract cations, which then promote nucleation. And so the viruses may be an essential part of triggering this whole mineralization process. Wouldn't that be something?
Yeah.
Yeah.
You wouldn't have travertine without viruses. I don't know how you could do an experiment to test that. That would be...
Yeah, I
could-
guess you'd have to make the biofilm in the lab with organisms either with or without phage.
You'd have to cure the phage.
Right.
And I assume people have done the purely inorganic experiment in the lab where you just take the calcium carbonate and heat it and try and make travertine. And I'm guessing that doesn't work very well. But yeah, as Brianne said, you'd have to make the biofilm without the phage. And I don't even know if you could. Some of these phages may be providing essential parts of that.
So you're sort of talking about alchemy of travertines.
Yes. Yes.
So if you've got travertine in your house, there's probably phage in there,
That's right.
Yeah.
Probably,
or at least fossil phage.
Fossil
a lot.
phage.
Yeah, a lot of bathrooms and kitchens have travertine, right?
Yep.
So,
One thing- Vincent, that you mentioned, and I see it in the text when they talk about figure 4b, but then a little bit further down in that paragraph, they talk more about it. Because in figure 4b, they list 2 enveloped viruses, the herpes viruses and baculoviruses, and they even draw them with a lipid bilayer around them.
They do.
So,
They do.
and they kind of later in that paragraph, then they mentioned that those are there. So it's kind of oddly written.
that's true. Yeah, they did find them. Although if you look at the colors,
They're not
Right,
abundant.
not a lot,
Well,
and they could be just contaminants, right?
And they found adenoviruses for me, and they found pox viruses for
Yeah.
Rich.
It's a little something for everybody.
Yep.
No picornaviruses.
No
No.
retroviruses.
No, they were all
for
DNA
Brianne.
viruses. Well,
All DNA
but
viruses.
maybe the RNA doesn't really survive. And did they
Yeah.
look?
Could
I
be
don't know that they did any RT
Right.
on these things.
Yeah.
Yeah, that's right. If you're just doing DNA sequencing, you're not going to get RNA viruses, right? Anyway, it's an interesting little idea that just another thing in the world is may be dependent on viruses, right? That's cool. All right,
Viruses rock.
viruses, right? It's very good.
Yeah.
It's very good. Viruses rock.
Or they're rock stars.
Viruses are rock stars. That's another good one. Oh my gosh.
You know, it took a while for geologists to accept the fact that there is a biogeochemical cycle for virtually every element below uranium
Hmm.
on the periodic table, and it took many years of experimentation and visiting places like South Africa, where they went down
Well,
into
time passes
gold-
differently for geologists. It was actually very fast.
That's right. That's right. When they went down 5 miles into a mine, I believe it's 5 miles down, for gold, and they started to look at this oozing stuff that came out of the side of the rock. It was microbial, so they call it snot rock. And they brought back samples, and they looked at it, and they were just astounded to realize that not only were the microbes in the rock, the microbes were eating the rock.
Yeah.
That was even more remarkable. I
Yeah.
heard a great presentation on this, and some guy from the University of Indiana had gone out to the hot springs and brought back lots of microbes, and also was studying the crust of the earth. And if you add up the amount of microbial life in the crust of the earth and put it on top of the earth, that I forget what the number was, but it was several miles thick of microbial life that you could engender by simply transposing it onto the surface and saying how much of it is there. It's coming out of the hot vents in the bottom of the ocean. That's very, very hot water. And it's not at the edges of it. It's deep down into almost the magma itself. So it's you're right. DNA is virtually everywhere. It's like Earth's biggest parasite.
It's life, Captain, but not as we know it.
That's exactly right. That's right.
All right, on to our paper.
Ah.
And this is a paper in immunity. Unusual to do an immunity paper here on TWiV, but you will see. And it's called Neutralizing Antibodies Evolve to Exploit Vulnerable Sites in the Hepatitis C Virus Envelope Glycoprotein E2 and Mediate Spontaneous Clearance of Infection by Frumento, Sinis Burosricas, Paul Stravakis, Zahid Wang, Ray, Fliak, Shaw, Cox, and Bailey. First author, Frumento. Corresponding author, Justin Bailey. Johns Hopkins, University of Bahrain, University of Pennsylvania, and Cornell University. Kathy's going to give us a summary of this.
I'm going to give a summary of the rationale, not actually what they did and what they found. So
you could do the whole thing and we could end it now.
Right. Hepatitis C infection is pretty serious. It can lead to liver cancer, other kinds of liver mortality, liver failure. And there is now an effective antiviral drug therapy for it, but hepatitis C infections are still increasing. So it would be really nice if there could be a good vaccine for it. But there are really serious challenges to making a vaccine for hepatitis C because there's so much genetic diversity in the hepatitis C realm. And in one individual, there are multiple forms of the virus that rapidly evolve. And in a sense, it's even more challenging than influenza. And so this paper comes about, or I learned from this, that there are individuals who clear the infection and then they clear subsequent infections 80% of the time. And so if the mechanism for this could be identified for this subsequent clearance, it could lead perhaps to design of vaccines to generate the same kind of response in everybody that seems to develop in these people that can clear the virus the next time they get hepatitis C infection.
So, Kathy, could you define for the listening public the difference between virus-free and clearance?
No.
Oh. Well, that's all for today, folks.
I wanted to hop in on just a couple of quick things here. So this is a little bit analogous to the elite controllers in HIV. except with hepatitis C, it's not so elite. It's about 25% of the people who get this virus spontaneously clear it. And
Well,
You can
Did-
get rid of it
it's- yeah,
100%. They're
yeah.
virus free.
they're virus free. I'm not sure if the elite controllers are entirely virus free.
right. So I'm just it's
Yeah.
it's
So
an analogy.
clearing it
yes.
from where? The blood, the liver, or
Well,
I
it's
gather you
probably
can't detect
probably
it.
PCR negative blood,
Yeah,
right?
so they're
That's what I'm
PCR
thinking.
negative and they don't have symptoms and they seem to have eliminated the virus. I mean, virus free, I don't know if you could ever really prove that.
they can't find the virus.
But
in other words.
the interesting thing, as Kathy said, about these people who do clear it is that 80% of the time when they get reinfected, they don't always get reinfected because hopefully, they're avoiding it. But if they do get reinfected, they've got an 80% chance of clearing it again.
Mm.
Whereas people who didn't clear it, obviously they still have it. And so the real question here is, what are the correlates of an effective immune response?
yeah,
We know that a fairly effective immune response can happen, which is really tantalizing. How does that work is the real question in this paper.
So I have a short addendum and a story I am compelled to relate.
Mm-hmm.
Single-stranded RNA virus envelope, right?
Yep.
It's
Plus strand, right?
plus strand. It's transmitted in modern populations mostly by transference of blood products or blood products. So intravenous drug use is a major vector. And back in the time before the blood bank was screened, transfusions were a big problem. And I have a relative who in the 60s, due to an issue, needed to be significantly transfused. And decades later, was shown to be, and the numbers here to me are astonishing, that it's 70 to 80% of individuals establish a persistent infection. Okay. And that's where the ultimate complications of liver cancer and cirrhosis and et cetera come from. Okay. So that's a big number. So this relative of mine discovered decades later that they were antibody positive in a routine screen for hepatitis C. And I said, dude, you need a PCR test. Okay. Because if you're antibody positive, that doesn't really tell you whether or not you have the virus. And there's like 70 or 80% chance that you still do, in which case there's trouble, but there's also treatment. Okay. And so this individual followed up on my advice, consulted their PCP, okay, who said, your brother's full of it. It's the other way around. It's 20% persistence, 80% clearance, with, complete assurance. Dude, you can look this up on Wikipedia. Okay, it's right there. So that's
or the
really
CDC
annoying.
website if you want another source. Yeah,
That's right. That's just so annoying when a doctor is so convincingly wrong. At any rate, they got the PCR test. They were negative. All good.
Good.
But that is a heads up. If through some routine test you wind up being, or if you've had a transfusion or were a drug user or something like that, you need to check this out.
Yep. Well, this is a study of antibodies in a cohort of people who inject drugs and different categories. So the name of this cohort is the Baltimore Before and After Acute Study of Hepatitis, or the BBASH cohort. Cute name, right? BBASH. And they're followed regularly with blood sampling, beginning when they were HCV seronegative. You catch them before they get infected. You can follow them through acute infection and then either clearance or chronic infection. And so you have this cohort who have, you follow them. It's really very, very good. It's very smart to have done this. And then they select some of the people out of this. They have people who spontaneously cleared at least one infection, eight people, or who had a single persistent HCV infection.
Yeah, this is a
8 of
good
those.
study design, but a very hard study design
Oh, yeah.
to implement because
It
you
is.
have to follow up with a group that's hard to keep track of and probably hard to win the trust of, and you
Yes.
have to follow up for years, repeatedly, and they end up with the 16 patients that they're going to talk
Right.
about here.
And the thing is, you have
so
to get
before you
them.
say, "Oh, that's a tiny number," this is really hard to get.
Right. Imagine that you had to get 8 out of that 20%.
Yeah.
Well, you also have to get them before they're Hep positive, and you have to convince them that they should be part of this study. And many of them probably say, "Well, I don't have Hep C. Why do I have to be part of this?" And you can't say you're going to get it, right? But that's what they do.
So, Rich, you said that there was a treatment. You were advising your relative that there was a treatment if they were positive. Aren't they obliged to treat people in this cohort that turned positive?
That's a good question. I don't know.
Let's see. What was the year here? Do we have that up front? It's a good question. Yes, you are obliged when someone
I know.
turns up.
Actually knew that answer.
What's the answer? You have to treat them.
You have to treat when you discover an infection that's treatable. You can't let them go on. That's the Tuskegee experiment.
No, you can't let it go on. So I don't know if this was long
this
ago.
This has got to be in the methods or the
This is an ancient retrospective study.
No, the really effective antivirals are more recent, right? So this could
in the methods.
Yep.
The second sentence under human participants says all samples were collected prior to the availability of direct-acting antiviral therapy for HCV, and none of the participants in the study pursued interferon-based HCV treatment.
Right.
Okay.
Yeah. There you go.
Good question, Dixon.
Thank you.
Yes.
Good. I'm glad you're listening.
I'm still awake.
The direct antivirals are fairly recent.
They look at the viral genomes. They amplify and sequence the viral genomes, and they're going to also look at the antibodies within those patients. So from
and
each
maybe
patient,
you said this, but we have time points, right? We have
time points.
Not only seronegative, but immediately after or shortly after getting the infection and down the road. And we have time points that include both what viruses they have,
correct,
and what antibodies they're making.
Exactly. So they look at the viruses and they can see that they're looking at the glycoprotein, which is in particular the E2 glycoprotein, which is going to be a target of neutralizing antibodies. And they can see lots of amino acid substitution over time. And they say in cleared infections, but not in persistent infections, the amino acid substitutions were enriched in the E2 part of the glycoprotein as opposed to the E2 glycoprotein as opposed to the rest of the genome. And they also look at evidence of positive selection. So in other words, if an amino acid change arises, does it remain throughout the sampling period or does it go away, in which case it probably didn't mean anything. And they say the cleared infections showed positive selection in E2, and most persistent infections didn't show any signs of positive selection. So in other words, the changes in E2 were maintained by selective pressure. They didn't just pop up there by chance.
And have you said that E2 is the virus attachment protein?
E2 is the virus attachment. I didn't say it. Said it was a glycoprotein, but yes.
Okay. So Vincent, that that's an interesting finding because you might suggest from epidemiological data that it's a subgroup of people that are more able to respond to this virus than the ones that are not able to respond. But in this case, it's the virus that changes its antigenic signature and then is more available for the immune system to attack it. Is that what's going on?
Well, okay, so they see this change. We're going to get to that. They do actually profile the virus later and determine that it's not that the people who cleared it got a defective virus.
Yeah.
there's something going on between the immune system and the virus that is more subtle than just win or loss.
We'll get to that, Dixon.
Yeah,
But hold
but
your question.
What they know from this is that the immune system in the people who clear it, probably the immune system, something in the people who clear it, is applying a selective pressure to the E2 glycoprotein, the main receptor binding glycoprotein of the virus.
I see.
And the people who establish persistent infections, you're not seeing the same sort of selection going on.
Right.
They're making antibodies, okay,
Not
but
against the E2.
it's a different situation.
Right.
So then they want to know about the antibodies in these patients, how they would neutralize the virus. So they make pseudoviruses.
Right, because the previous thing kind of implies,
Yes.
hey, maybe the thing that's inducing this selective pressure is some really good antibody.
Yeah, because you see changes in E2. Yeah, maybe antibodies are selecting that. So they take the glycoprotein and put it into another virus, so they don't have to use Hep C, which is very difficult to grow, and you can't do plaque assays and neutralization assays. So they're called pseudoparticles, but they can introduce the amino acid changes into those. And for people who had cleared infection, their early plasma was able to neutralize early. They have both E1 and E2 and the whole glycoprotein complex. But the late E1, E2 from the same infection were resistant by neutralization to the early plasma. Okay?
So when you make those changes in E2, they're no longer can be neutralized by the plasma that you made it to start with.
That's right.
Right.
That's right.
Right. So these appear to be escape mutants.
Yes.
There was an initial immune response. It produced antibodies that were effective at binding this protein. The virus has developed mutations that allow it to avoid being bound by that antibody, which is pretty much what you expect, but it's happening only in these patients who successfully cleared it. So then the question becomes, well, how did they clear it if the virus is escaping those antibodies?
Exactly. So, and then they narrow it down to E2. The substitutions in E2 seen in the cleared infections were probably selected by early plasma neutralizing antibodies. And then the late post-clearance plasma has the capacity to neutralize late contemporaneous E1, E2. So in other words, the neutralizing antibody response evolved to be able to neutralize the variant virus that was previously selected.
Right. So we made some antibodies that could neutralize early virus. The virus changed.
Right.
Antibodies also changed.
Right.
And now those new antibodies were able to deal with this
deal with the virus and
Right.
the cleared infections. But in the persistent infections, there was minimal neutralization by either late or early plasma. So there's not any selection of variants in the people who could not clear their infections.
So, Brianne, in your mind, are those late antibodies, are they new antibodies or are they evolved antibodies?
I could imagine that actually being either. I actually was staying out of that when I described things earlier because we don't know, is it a new B cell that is now participating in the response, or is it some extensive hypermutation on the original B cell?
Is that a new B or a newbie?
Right.
and your perspective is it could be either.
My perspective, at least at this point, is that it could be either.
Okay.
Mm-hmm.
What is an evolved antibody?
An evolved antibody?
Yeah.
I'm thinking of what's it called? Hypermutation.
Somatic hypermutation.
Somatic
So an antibody
hypermutation.
that's made by a B cell that has seen antigen a lot of times and that has undergone some somatic hypermutation in the germinal center. So it's made changes to adapt and better bind to that antigen.
Right.
Okay.
So they want to know what these amino acid changes in E2 are doing. And the first thing they think of is, let's look at binding to the receptor, because we know that antibodies can block receptor attachment to cells. So there are two receptors for hepatitis C virus, CD81 and scavenger receptor B1. Such a great name,
Yeah,
scavenger.
scavenger receptor, right?
So 2 different cell surface receptors. And they do binding assays. They find a decrease in binding to CD81 from early to late for cleared infections. Okay, so people who clear the infection from the early isolates of the E2 to the late, you see a decrease in binding to CD81. But from persistent infection, there's no change in binding over time. Okay.
So these
and
changes in E2 exerted a fitness cost, probably,
That's
on the
right.
virus. It doesn't bind as well to its receptor after making these mutations that allow it to evade the early antibodies.
Right. So if you make the antibodies, you sort of push the virus into a less fit
Which is
configuration.
very interesting that the only way you can get away from the antibodies is to become less fit, right?
Right.
It's very interesting. And you know what? We talked about fitness last time, and I'm glad this is all about fitness because, in the Marek's paper, I
Well,
was
it's
ranting
the
about
new
fitness.
year. Everybody should be about fitness, Vincent. Come on.
Yes.
But I think it's interesting that the escape changes confer a loss of fitness.
And I would guess that both the new/matured antibodies and the fitness cost contribute to the clearance.
Probably.
Yes.
Yes, absolutely. Okay, and then we do the experiment that Alan had mentioned a long time ago, months ago. No, earlier. They wanted to see if there's a difference in binding to CD81 between the cleared and persistent infections, and they find no difference between the two groups, indicating that the people who cleared infections were not infected with a defective virus compared to those with persistent infections. Right, the difference in outcome is driven by differences in the immune response, right? Selection.
And this is with the early virus.
Yes.
Yes.
Not with the late virus. Late virus.
Early
Right.
has
Late virus
changed.
has a defect, is not as effective at binding CD81, but the one they initially got, the early virus, is effective at binding CD81.
So that
it
was the
didn't
answer
come
to my
in broken.
question then. Yeah. Okay.
Yeah.
Good. Very good.
Then they want to look at specific amino acid changes that are involved in this phenotype that we've been talking about. So they introduce individual changes, single amino acid changes into E2. And they say most E2 substitutions from cleared infection resulted in reduced CD81 binding. Okay, confirming what we already saw with just the whole E2. And some of the persistent infections had amino acid changes that reduced binding, but most of them did not. So it's not black and white. Okay, not all of them had no effect, but most of them did not. And so they call this is very nice. It's very timely. Decreased the changes in E2, the amino acid changes that lead to decreased or increased binding is loss of function or gain of function. Okay, so decreased binding is loss of function, which nobody mentions these, right? Everyone else talks about gain of function, but that is the increased CD81 binding, loss or gain of function. Okay, and then they said they introduced each of these amino acid changes that they have just looked at in terms of CD81 binding. They look at them in terms of neutralization. And they have 4 neutralizing antibodies with 4 different names, of course, in there. So they have them in both their inferred, reverted, unmutated ancestor form and their mature form. How do they do that, Brianne?
So they know that each antibody is made by a combination of a V, D, and a J gene segment. And we know what the sequences of those are in non-B cells.
Right.
And then we can compare what they are in these mutated B cells to see what kinds of mutations have happened.
Got it.
And this kind of gets at that question that Rich asked, because you can make those comparisons and see if these are the same B cells from the same lineage. They chose the same V, D, and J and then just made some edits. You can see that they're the same B cells doing hypermutation and editing, but you have to actually look at the VD and J sequences to do that.
So they have the inferred antibodies that were present at the beginning of the infection, right? And then the ones at the end with either clearance or persistence, depending on what group of patients. And those have a bunch of amino acid changes in the antibodies that are changing its property, right? As we will see. Okay, so most loss of function and gain of function substitutions gave increased resistance to most of the unmutated neutralizing antibodies, all right? But increased sensitivity to the mature, late, hypermutated antibodies, all right? So either the loss or gain, which is loss of binding, decrease in binding, or gain, increase in binding, those had increased resistance to the early antibodies, increased sensitivity to the later ones. So for each, they have antibody pairs, right? An early and a mutated form. Each loss of function conferred resistance to the early form and increased sensitivity to the mature form, and the set of the same for gain of functions. Gain of function, increased binding as well. And then they map these on the structure of the antibody, and they find that the loss of function changes. So the ones that decrease receptor binding, they decrease the fitness of the virus, or all over E2, and they're not near the place where antibody binds, and they're not near the place where CD81 binds either. So
it seems like there are multiple ways for the virus to get away from the antibody that all reduce its ability to bind to CD81, but they get it away from the antibody.
And all these changes are not near where either the receptor
Not
or the
near the
antibody
binding
binds,
site.
which is interesting. So they must involve some major conformational changes. Who knows what, right?
So if you had a monoclonal antibody against these changes, you could give them to a patient who could not respond to the virus, and they suddenly could get rid of their virus.
Well, I think
Is that a treatment?
I think you need a vaccine that would induce
Yeah.
these antibodies, right?
Yeah, what
Okay.
they're getting at here
Okay.
is you want a vaccine that will induce this type of response.
You want the late antibodies, right?
Yeah, but
Yeah.
without a vaccine,
no, you want the
still have a
early
treatment.
antibodies.
You want
But
the
the early
early
antibodies to select for those loss of fitness,
to
Yes.
push the virus.
and then you also need the late ones to
Yeah.
be there as well, right?
Yeah, because this is so it's like the virus is playing checkers and the immune system's playing chess. So the virus mutates. I escaped your antibody. Yeah, right into my trap. Right. So you've got to get both steps of that process, because the late antibodies aren't going to neutralize the early virus. They need the late virus that you get from the early antibodies. So you need that whole panel.
So they say a vaccine, a vaccine could induce these kinds of antibodies. They would induce a mixture of immature and mature antibodies. The immature antibodies might select the substitutions in E2, and the mature ones would then neutralize them.
Yeah.
More effectively, right? That's what you need from a vaccine. Good luck.
Is there a CRISPR approach to this?
You mean
Probably
creating
engineering the
individuals who
human?
can make
That's
those
right.
antibodies?
I mean, you've got the sequence for the VJ regions and the J regions. Why can't you just engineer that into the bone marrow?
Well, then you'd have 100% of your antibodies binding to
I don't mean
hepatitis.
100. Well, I see.
So
But what about the people who do have the response? Are they 100%?
No,
Well,
but I think
so-
if you're it depends on if you're engineering the bone marrow or not.
Right. I think technologically we are more likely to get to a vaccine that induces this response a whole lot sooner than we are to get to a CRISPR approach that would be not completely disruptive of everything else.
So
my question is now, well, why is it that those patients made these antibodies while the
Yes.
others did not?
Bingo.
Yeah.
right.
And
Bingo.
how can we start to push more people towards
well.
making those
And
antibodies?
in fact, you may not have to have a vaccine that produces both sets of antibodies. If you have a vaccine that produces the first set of antibodies very
Mhm.
effectively, and then the virus comes in, you select immediately for the mutants,
Right.
you may then induce the production of these secondary antibodies. But this
Yeah.
is a first step toward figuring out how this clearance process works. And obviously, there's more that needs to be done before we actually get
Right.
to a vaccine. But this is a really,
But
I think, a major breakthrough.
but Briam's question is right, because not everybody makes these antibodies,
Yes.
right?
Right.
So, Briam, what controls how you respond to an antigen?
Come on, Brianna, you've got all the answers.
Let's see. How much time do I have?
Is
for,
it T
you know,
cell? I mean,
No,
is it
no, no, no,
MHC?
no.
Is it T cell receptor?
It's probably T cell receptor and MHC. It's
Hmm.
probably binding strength to antigen, and so
Yeah.
amount of signal. It's likely to be quite a few different things, is my guess. But even the knowledge of, we have to figure out how you make this specific kind of antibody because this antibody is key,
Yeah.
is a good, part to make this start.
Right.
So what my main question is it possible that these individuals the 80% who make persistent infections are actually genetically incapable of making the right profile of antibodies?
What do you mean by genetically incapable?
Well,
Are you getting at,
so
is
you
this-
have to.
Is this an HLA haplotype issue?
Yes,
Yeah,
exactly.
Okay.
So maybe they can't present the T cell, the antigen, to get the T cell help for this. I think we'd have to know a lot more to figure that out.
I think we need a lot more than 16 patients to get at that.
Yeah.
So my other question is, and I've never really, actually I have a bunch of questions, but here's one that I've never really fully understood. If we have a virus here that is capable of a lot of mutation, okay? And yet, over time, I mean, you can describe a genome, right, with a sequence. Is there a bottleneck somewhere so that you all start off with the same thing and then there's variation, but only a subset of those, a select few, can make it to the next host?
Yeah, I kind of think about it that you have the virus with sort of its original sequence,
Right.
and all possible mutations are being made, or all possible changes to the nucleic acid are being made, but some of those are not going to be fit.
Right.
And are going to be selected against. And so in the end, you get almost like the most fit or best sequence structurally, which is kind of the sequence at the end.
So I don't know if the part of this question is, could these individuals who are either clear the infection or persistent, could they have gotten slightly different viruses to start with? Yeah, I don't even know if that's addressed in the paper. So
Well,
that
in terms of the binding, no, they did address that.
Right.
Right.
It's not, they're not different in terms of CD81 binding,
yeah.
but could they have been different in some other way?
Sure.
Maybe.
Sure. Probably were. Right. Yeah.
Yeah. Yeah, it's hepatitis C, so everybody gets a different virus, but they're all built around the consensus, to make it hepatitis C.
Yeah, and how many, they have 8 here from each group.
Yeah.
So they have 8 sequences.
Yeah.
That you'd probably need a lot more than 8 sequences per group to decide if there was some kind of consensus change.
Right. Although it did just occur to me, one strategy for developing a vaccine in this, backing up a little bit, would be to use a live virus. Sorry, Vincent, an actively replicating vaccine, so that the vaccine pseudotype virus would go through this same transition, and you could potentially get the early and the late response, maybe.
But you have to know what to start with, right?
Yeah, you got to know what to start with, and you've got to know that it'll predictably do that in every patient you give it to, and that's a ways off. But this is a first step.
As Alan said, this is important because now we understand why this fraction of people are able to clear infection and now can move forward, because we need a vaccine. The drugs are not enough.
Yeah.
Right.
Yeah.
Just listening to this conversation as if I didn't know what you were talking about. If you hadn't mentioned the word virus, I would have sworn you were talking about African trypanosomiasis, because it undergoes similar
antigenic
changes,
switching.
And it's a little bit different, of course, because it's more complex, but the result is the same. Ultimately, you die without the drug.
I think in the end, a lot of this is coming down to kind of the biochemistry of structural constraints.
Yeah.
Yes, that's it. Okay, let's do some email.
Love it.
Great paper.
Yeah, very interesting.
I
want
agree with
to.
you.
I will take the first one. It has to do with polio. Yeah, Beth sent this to Daniel for clinical update, but I decided we should do it here as a little. We're going to do it here. Beth writes, I followed with interest the discussions about polio in the weekly clinical updates and TWiV, because we also talk about this a lot on TWiV. It seems to me that given the huge number of people in the New York City area, the constant influx of international travelers, and the likelihood of oral polio vaccine resulting in contagious infections, that vaccine-derived polio should be actively circulating in the metro area most of the time. Yet, the municipal wastewater analysis page reports only one positive concerning sample in Rockland County in 2024. Does that mean that the wastewater surveillance system is not capturing a complete picture, or is it more or less accurate and polio is not currently circulating in New York? At TLDR, not capturing a complete picture. Okay.
You don't see what you're not looking for.
Here's my answer. I wrote it out. What is a negative result? It doesn't mean that there's no polio RNA in the sample. It means the amount of polio RNA is beneath the level of detection. Okay? Very important point. Now, wastewater sampling for detecting polio is very controlled and restricted because the WHO, it's based on the idea that wild-type polio 2 has been eradicated, as declared by WHO in 2016, and I've talked about this on TWiV several times. So, to prevent the reintroduction of wild-type polio 2 in the world, WHO has issued guidelines that stipulate that any form of type 2 polio must be handled, any form of type 2, any form must be handled and transported under the Global Action Plan for Polio Containment Guidelines, or GAP 3.4, GAP, Global Action Plan, okay? This document outlines procedures required to address and maintain the biosecurity and biocontainment of type 2 polioviruses. For instance, anyone exposed or infected with type 2 polio must be quarantined, and their stool must be collected and tested for the presence of infectious virus. You may remember we talked about the case in the Netherlands where someone working in a polio lab started shedding polio. He had to be quarantined for a month over the holidays, remember? He had to get psychological counseling to make sure he was okay being alone over the holidays. I would like to be alone over the holidays.
Yeah, it depends on their family situation.
For instance, yes, quarantine. Only after 2 consecutive stool tests are negative is the person taken out of quarantine. It's also because of these guidelines that it can never be proven that the administration of remdesivir cured the chronic excreter. Remember, we talked about that as well, as the last stool samples could not be transported to the appropriate facility for analysis. So in other words, the pre-remdesivir, this is a person who is excreting poliovirus for many years. He was given remdesivir to treat COVID, and he wasn't excreting any more polio, but they had no pre-remdesivir sample because of these guidelines. You couldn't ship type 2 anymore. You couldn't ship type 2 containing poop. Okay. Now, the U.S. has agreed to follow these FRCOCTA guidelines. I'm sorry, these guidelines.
FRCOCTA is a virological term that you can look up.
So there's several issues. First of all, how do you place a community under containment if type 2 polio is found in the environment? Now, the guidelines in the GAP documents can be applied to labs and lab workers. They cannot be applied to geographical areas or large populations, obviously. So this had not been thought through
outside
properly.
of the Simpsons movie.
Furthermore, since most polio infections are asymptomatic, it's very hard to identify patient 0. So, with these caveats in mind, the U.S. CDC has decided that wastewater sampling should only be performed in community with known transmission of poliovirus. Transmission means a case of paralytic polio. Therefore, there's no wastewater sampling in most entry points in the U.S. In 2022, wastewater sampling was done in communities known to associate with the Jewish community in Rockland, where that patient was identified. And that explains why Dutchess County was tested, the wastewater from Dutchess County. This policy also says that wastewater sampling can stop after 6 weeks of negative sampling results. So few wastewater sheds are continuously tested or tested at all for the presence of poliovirus. Okay, Beth?
Vincent, when I was reading the this email, I had perhaps a much less informed thought.
Yes, it's okay.
But fortunately, I know exactly who to ask my question to, which is that Beth's assumption is that any of these international travelers or these large number of international travelers are going to be shedding oral polio vaccine.
Yeah, that's right.
So my question is, are they shedding for a long period of time after having been vaccinated? And do we think that there are a lot of people who are traveling in the time window during which they would be shedding?
Yeah, they can shed for weeks for sure. Okay. Even months. I mean, Phil Miner's kid shed for months, and his kid was fine. He had no immunocompromise. So, but the point is that we're not testing because there's no case of polio. So there's no polio transmission now. So we're not looking.
Are there not population centers elsewhere on the planet similar to New York where there is regular wastewater
There's no place
testing
like
done?
New York.
Yes,
Okay.
That's why before that summer polio case in New York, they found it in London sewer. They found the same isolate, and before that, it was in Israel sewage. So yeah, it's all over the place. Similar things happening. Countries using IPV.
Is there consistent sampling in these places so that you can see sort of the prevalence over time?
Any place that follows the WHO recommendations on containment, which is going to be pretty much the industrialized world that's capable of doing this type of sampling on a regular basis, is going to encounter this same thing and is probably going to not test.
Right. Okay, so there you go, Beth. Thank you very much.
Yeah.
Kathy, can you take John?
Yes, Doctor's TWiV. As of this week, I can now count 4 people I've known, one quite well, that are now COVID stats. The virus got the first 2 before there were any vaccines. The latter 2, I suspect, were unvaccinated, if not outright anti-vaxxers. Also this week, my fully vaccinated son got it for the second time. As an attorney with the public defender's office, he's probably often in the presence of the less than vaccinated. That aside, he's feeling better now, but texted me that he'd hate to imagine how he would feel if he hadn't been vaccinated. And with that, this popped into my head. Remember Cy Sims, the purveyor of surplus stock men's clothing warehouses? The slogan was, "An educated consumer is our best customer." So to paraphrase Cy Sims, I replied to my son, "An educated immune system is your best friend." Otherwise, in 1077, besides the science, I was quite interested in hearing Sana Naderi's story and how it is in Iran. Thanks for asking her about that. Best wishes from a gray Greater Braddock that got little snow from yesterday's event and stayed between 33 and 36 Fahrenheit all day. John.
You like that saying, an educated immune system is your best friend, Brian? Is that good?
I do like that
That's
saying.
good.
Okay.
I think I should put that on top of my immunology syllabus.
And I agree. It was interesting to hear about the science in Iran. I was glad you asked that question also.
I was just sitting there listening to her, and I thought I should ask her this. Yeah, it just popped into my head. Alan, could you take the next one, please?
Sure. Michael writes, I recently listened to episode 1075 about Marek's disease virus in chickens. As discussed in the episode, the virus became more pathogenic in the early 1900s after the advent of industrial chicken farming, and the higher pathogenicity is associated with higher viral replication. It seems to me that in pre-industrial farming, a faster replicating but more pathogenic virus would have had a higher chance of killing the small flock it infected before being moved into another flock. But the rise of industrial farms with thousands of birds crammed into small spaces opened a new niche for faster replicating, more pathogenic viruses. It didn't matter that they killed their hosts faster because there were so many hosts around. I'm not sure how you would test that hypothesis, but for the sake of argument, let's assume it's true. Around the 40-minute mark, Vincent states that a less fit virus is one that doesn't replicate as much and that, quote, over time you select for higher viral replication. But if it's true that industrial farming created a new niche for faster replicating viruses, that's not entirely the case, because faster replication is associated with higher virulence in the host in this particular virus. The faster replicating viruses are only more fit in the new niche created by industrial farming. Thank you, Rich. You were correcting that typo, right? Yeah. For those who are not regular listeners, I suffer from a condition I call proofreader's eye, and my mind stutters when I see something that's a typo. And if you somehow transported a modern high-virulence virus to the past, it would kill too many hosts and burn out before it could become more widely established. In that pre-industrial environment, a faster replicating but more pathogenic virus is more fit from an in-host viral replication standpoint, but it's less fit from an evolutionary standpoint because it's too virulent to efficiently move between host populations. I'm not a virologist, but it seems weird that fitness is just focused on viral replication without taking into consideration how that replication affects a virus's ability to infect new hosts. A virus that kills its hosts too quickly to spread seems less fit regardless of how many copies it makes of itself. And Michael is an assistant research professor of arthropod identification at Penn State. Very cool.
So the assumption is that if you kill too quickly, you don't transmit. And I think that's an incorrect assumption, because you can transmit an awful lot before you die, no matter how quickly.
It depends on how you're transmitted.
Yeah. I mean, I think Ebola patients transmit quite well, even though they get killed relatively quickly. So I think we assume that that's the case, but I'm not so sure. And so there are a lot of assumptions in this letter. The other one is that the faster replicating viruses are only more fit in the niche created by industrial farming. You don't know that. That's an assumption that may not be correct. So, but I think the crux here is that I'm not convinced that killing faster makes any difference. I think you can still transmit. So
Crows get sick, 2 days later they die, and in between time they transmit West Nile virus.
I would argue that it matters a lot how the virus is transmitted. The reason the sick crows can transmit the virus is because I mean they're and you're talking about a mosquito spread disease.
No. This is crow to crow. I'm sorry. This is not
crow
mosquito.
to crow. Okay. Okay. Well, yeah. Okay, all right. Highly social animal.
Very social.
Yeah.
Even though they do murder, they're highly social.
That's true.
Yes,
we
they
happen to
murder
be
all the
one of
time.
those social animals, by the way, so don't forget that.
So, Alan, give me an example of a virus that would be affected, in your opinion.
A respiratory virus. If it replicates to too high a titer and the host doesn't get out and interact with other hosts, then you got a problem.
Yeah, but what if
versus
there's
a vector-borne virus where the host can be completely incapacitated and the vectors can still feed on them and spread?
Right, but even with these respiratory infections, there can be an asymptomatic period of shedding before symptoms, so you're going to transmit it.
If you replicated to a higher level, you probably wouldn't have a long enough asymptomatic period.
I'm not sure that's correct. I think you could do that and still be asymptomatic.
I can't agree that it's all about replicating to a high titer. I mean, look at all the viruses that do very well without doing that, like herpes viruses.
Well, they're not very fit, obviously.
Student versus mentor.
Except that everybody has them, right? I mean, that's a highly fit virus.
But when I say higher replication, there can be many reasons for that, all right? There could be evasion of immunity could lead to higher replication. So he's saying that it's only one thing, but it's not really only one thing. I use that as a catch-all for many different mechanisms. So.
How is Merrick's transmitted?
Kissing, feathers, right? That's
feathers.
What
feathers. That's right, feathers.
But
Marx Brothers.
It's acquired as a respiratory infection, right? It's carried on feathers. The feathers are a fomite. Preening. You got to have one chicken preening another. Do they do that?
I think
it's
they might.
Chickens don't give a shit about each other.
No. It's a pecking order, goddammit.
It's transmitted through the dusk and dander, so they must inhale it. They must.
Right, but which also, in theory, would mean that the chicken doesn't have to be alive.
No, it doesn't have to be alive. That's right. In fact, I think Angela said you can have a feather. You pull a feather out of the chicken and it can transmit. Okay.
What I take away from this letter.
So they come down with the virus.
They come down with it. Yeah, I got that. Oh boy.
What I take away from this letter is that, the definition of or the description of fitness could be makes sense to me, sort of dependent on the niche you're talking about. And here we're talking about
sure.
wildly different niches between a natural chicken environment and industrial chicken farming.
Well, and even fitness in cell culture versus fitness in a full organism.
Yeah.
Or
sure.
Or fitness in transmission among a population.
I don't think you can assume it's different. You have to show it, okay?
Yes.
And Brian, you turned away from your mic because suddenly you got quiet.
No?
Okay.
I'm back.
Who's next? Did you go, Kathy? Yes, you did. Rich, you're next.
Matt writes, first off, big fan of the TWiV podcast. I was turned on to TWiV by a random swim parent who said early in the pandemic, "You sound like these guys I listen to every week. Have you ever heard of TWiV?" She didn't understand half of what you said, but learned a lot anyway.
That's me too.
I'm an NIH researcher, cardiovascular icosanoids mostly. And I enjoy the In the Weeds coverage weekly. We've actually tested a drug to battle the host response phase of COVID, but we kind of had one shot at it, and after seeing the data, realized how bad a model the K18 HACE2 mouse really is. Way too harsh. Good for vaccine and mRNA analog testing. Bad if you think you might have something that offers mild improvement, in my opinion. I typically hate hearing my words read back on a podcast, but I have a question I'm not sure anyone knows the answer to and may be of general interest. Maybe you could paraphrase. Not a chance. I'm going for it. My son caught some crud that some would call a common cold before Christmas. No diagnosis of the crud. Non-COVID per antibody test. He had got a flu shot this year. It seemed viral, not bacterial. 2+ weeks later, he still has a moderate lingering cough. To me, the COVID data was revelatory in that the host response phase isn't just after the viral phase. It seems post-viral, minimal remaining virus, minimum contagiousness. In human ACE2 mice, viral copies drop 100-fold from day 2 to day 4, and mice start dying of practically sterile pneumonia on day 6 or 7. I'm wondering if they have any idea about the length of the Vincent, you're in my way. Length of the viral phase versus host response phase in other viruses. Are there any data or a sensible common rule, 5 days, of isolation or masking following a common cold, flu, etc., before one emerges back in the world? I sent him back to school 13 days after the first symptoms, and I feel comfortable with that despite mild coughing. Another son and my wife are on a similar trajectory. Was I wrong to send him back to school? Sorry if you've covered this. I've only listened to the past 200 shows or so. So, to paraphrase, if I can, the question is, are you typically virus-free and non-contagious before your symptoms have all cleared up?
Mm-hmm.
And if so, what's the timeline? And if so, is the timeline the same or different for different viruses? Okay. I don't know the answer to that.
It's different.
My assumption is that your symptoms linger significantly in general after you've cleared the virus. Okay.
Yeah.
Whether that is the same or different for different virus infections, I wouldn't know. My guess is there's
it goes below it goes below the transmission level.
Right.
You may still be shedding virus, but it might not be enough
that's right.
to infect another person.
I would assume that there is some variation from virus to virus, as well as some variation from person to person.
Yeah.
Absolutely.
Right.
I mean,
Yes.
very typically rhinoviruses you shed for 4 or 5 days, and then you have a long tail of sore throat and coughing and just annoying as hell. 2 weeks.
Right.
Yeah,
So
I think flu basically goes down to baseline within about a week of the
Mhm.
start of the infection, but your symptoms can persist significantly beyond
Well,
that.
COVID does that too.
Yeah.
Yes.
Right.
So that's what
So
he's
the point,
comparing
yeah,
it to.
that's
Yeah.
the
Yeah,
point.
that's
And-
right.
The
And
COVID
in-
model applies.
And in all of those cases, when we say it goes down to baseline, I would say that that really means it goes down to a very low probability of transmission, but not zero.
Right.
Right.
So
Yeah,
I don't
okay.
think you were wrong to send your kid back to school. 13
No.
days sounds like a lot.
That's a lot of time off school.
That's a lot of time.
True.
Boy, he missed a lot.
He did.
Or drove the parents
all right,
crazy
that
for a
ble-
long
that-
time.
Who knows? Maybe he was listening to back issues of TWiV, in
Could
which
be.
case
That's
he
right.
did
You
fine.
he was binging.
All right. So one more from Brienne.
All right. Jonathan writes, traveling to rural Africa and just got the recommended 2-dose rabies pre-exposure shots, but can't get a straight answer from my doctor on the true benefits. She said it reduces the severity of the disease, but since the ultimate symptom of rabies is almost always death, does that mean that the PrEP is sufficient on its own to render rabies survivable sans post-exposure treatment? She said I should seek treatment ASAP after getting an exposure, which seems logical, but since I'd be doing that anyway, what is the benefit of getting the PrEP at all?
There's a
Thank
big
you,
difference
Jonathan.
between nearly dead and dead.
And all dead.
And all.
Yeah.
Well, you may be somewhere where you can't get rabies prophylaxis, right? And what they do is if you get bitten, they'll inject monoclonals at the bite site and then vaccinate you. But maybe you're somewhere where you can't get that or they won't do it. You might as well be prepared, right?
Well, my wife and I went on safari about a year ago to rural Africa, 3 different countries, and it was not recommended to do that. Just the malaria prophylaxis.
It
And
depends if rabies is endemic. If you go to India, you should get a rabies shot
Oh
because
my
there's
gosh, and
70,000 cases a year of rabies
that's
in
right.
India.
No
right?
question. No question.
But I would definitely do it. It prevents you from dying. Yes.
Yeah.
It's a very good
As it
vaccine.
does, it does.
And with rabies, if you're unvaccinated, 99.9% fatality rate.
Yeah, when there are things that are pretty serious fatality rate, I'm okay with being covered 2 different ways.
Yeah.
Sure. Okay, Dixon, let's do some picks.
Okay, since I didn't read any mail, I can do 2 picks.
Oh, I forgot. Sorry, Dixon.
Yeah, that's okay. I live for those moments, though. I really enjoy reading whenever I can.
Do you want to let Dixon read an email?
No,
Yeah,
no,
if you-
no, no,
you
no,
can
no.
read an
Don't
email.
don't do that. Don't do that.
Okay.
So I have 2 picks. I have one as a confirmation, as if we needed it, from the European consortium that calls itself Copernicus, and they monitor the environment. And they concluded 2023 was the hottest on record. Surprise, surprise. And I heard a depressing NPR story the other day from a climatologist that says if you live long enough to look back at 2023, it may well have been the coolest year of your life. I didn't need to hear that, actually. And then I thought some cheer because obviously we're into the new year. Here are 7 songs that the New York Times has rendered for us through something called Amplifier, I believe. And I haven't listened to any of them yet, so I can't guarantee that they will strike a accordant note in your psyche. But they weren't wrong about the 20 that I did recommend before, so I think these are probably pretty good too. Worth a shot and have fun.
All right. I put the right link in there.
That's right. That's right. That is the right one. That's the
Take out your Gmail link.
Thank you.
Thank you, Dixon.
You're welcome.
Brianne, what do you have for us?
As I'm sure you all know, I have spent some time in the past couple of years thinking about trivia. And a lot of people who I know have told me about different ways to learn about trivia, one is which of which is some trivia podcasts. So I listen from time to time to one called Stuff You Should Know while driving. And I listened to an episode that was adjacent to things I had read about in science fiction, but not a specific topic I'd thought about. I found it really, really fascinating, which was MEDI. And it is related to a program people may have heard about called SETI, or the Search for Extraterrestrial Intelligence. They also have an episode on SETI and just sort of the history of looking for broadcasts and what a broadcast would look like and sort of the science of that, which I found really interesting. But then I wasn't as familiar with the fact that we also have programs in MEDI, or whether or not to send a message ourselves, what these messages should look like, and very important philosophical and ethical debates on whether we should do so. And I hadn't really thought about a lot of these issues before. And I was listening to this the MEDI episode while driving, and I went and found the SETI episode right afterwards, because I learned a lot that I hadn't thought about with these things. And they were just really interesting things to think through.
So, Brianne, haven't the horses left the barn already on that one?
So the way that they discuss it is that the horses have left the barn in a very local area, but that some of the signal has degraded out to areas that might be also fruitful. But though they do also talk about the Voyager record that was sent.
Right. The Voyager record, and also every radio transmission that's ever been
I
sent.
was
on
going to
Earth
say, and virtually every radio show
is irradiating space in the local environment, but our total wattage decays over time as the signal decays over distance. And it's likely that on distant systems, actually, you don't even have to go that far before it's not going to be receivable or it's detectable as something produced by intelligent life versus just radioactivity, especially since we have so many signals overlapping. So I think that a lot of the MEDI debate, a lot of the MEDI debate centers around, should we try to send higher powered signals out, and might that be a bad idea?
It was just so much of a more well thought out and organized debate than I had really imagined. That there had been that much thought put into it beforehand. I thought it was interesting.
Here's a good quote from Wikipedia on this. Science fiction author David Brin has questioned, quote, whether small groups of zealots should bypass all institutions, peer critique, risk appraisal, or public opinion to shout yoo-hoo into a potentially hazardous cosmos.
That is, in fact, also discussed in the episode.
That's good. I like that. That's really good.
This reminds me of that bit with the high-end physicists. This is the Fermi thing?
The Fermi paradox.
Paradox.
This is related.
Where is everybody?
Right. Why haven't we detected any other intelligent life out of all of the billions and billions of other systems? Is it because it's never happened, or is it because everybody else knows not to say anything?
That's right.
That's exact. All of these things are exactly what these episodes were about. So if you're looking for something while you're driving, I found it really interesting.
Good. All right.
Very cool.
Thank you.
Thank you. Kathy, what do you have for us?
Well, I have several picks about Oppenheimer, but first of all, it kind of stems from the fact that in 5.5 years ago, I went to Los Alamos and visited Bathtub Row and toured things. And that was the summer when the opera was John Adams' Dr. Atomic. So it just seemed really fitting. And I went again this past summer when, of course, the movie about Oppenheimer came out. So it seemed really fitting. And I saw the Oppenheimer movie twice. And Rich had a pick about an Oppenheimer bio subsequent to that, or sometime around that time. But I was in Mike Imperiali's office, and I noticed that he had this magazine that was the Los Alamos National Laboratory magazine called National Security Science, and it was the summer 2023 Oppenheimer issue. And so I said, hey, Mike, I'd like to borrow this. And I read it cover to cover, which took me a long time, but eventually I found that there's a PDF. And so I put that link in here for you if you want to get more info about Oppenheimer. But the other thing that came out from reading the articles in that was that Los Alamos lab put together 3 documentaries, each 27 minutes long, about science, mission, and legacy. And they're quite well done. And so I put the links for those in here as well. So if you want more about Oppenheimer and Los Alamos and so forth,
Cool.
there you go.
So, Kathy, the security issues surrounding the development of the atomic bomb were heroic and resulted in 2 people losing their lives because they thought they were spies, right? The Rosenbergs. And how did the other countries come by their atomic bombs if they didn't steal them?
Oh, they did steal them.
And
It's
who
well documented.
gave them
There's
that
a
information?
there is a
many of the sources were people at Los Alamos who
Yes.
were leaking information to the Soviet Union.
These are physicists now. These
Yes.
had to be
There are
physicists.
at least 2.
there,
that
yeah.
there's
there was
And we
evidence
know
as-
their names, and we know their names.
Yes.
Mhm.
as is
Right.
pointed out in both the movie and the book. There was a school of thought that we should share everything. Even Oppenheimer
Yeah.
thought
sure.
so
Right.
for a long time. That the
Yep.
way to keep the playing field level and therefore sort
level.
of quench the
Do you
fire
mean
was if
level?
everybody had the technology.
Right. The idea was to what Rich just said. If everybody had this technology, then nobody would use it.
Or they could level the field, and quite
Right,
literally.
right, in the literal sense. Yes.
The
You remember when Niels Bohr went to Churchill to propose that? Churchill said, "Get out of here," right?
This is dealt with a little bit in The Making of the Atomic Bomb by Richard Rhodes, and
Yeah.
even more so in the sequel, Dark Sun, about the hydrogen bomb by the same author. The first part of Dark Sun was about the Russians' development of the bomb and about how basically they copied it from the Americans. And a lot of that information, I forget who was responsible for this, but was transported in diplomatic pouches secretly between the two countries, because that stuff is not really censored. Okay.
It
Great.
wasn't Michael Pollard.
Wow. Amazing. History is amazing. Rich, what do you have for us?
Okay. In the we do have a sort of a sailing arc in our science
You
picks,
do.
even if it's
Yes.
not science. I hope this has not been picked before, but I don't care because I'm going to do it again. I actually saw this book previewed on PBS NewsHour as one of the best of the year, and then my son gave it to me for Christmas. The book called The Wager, A Tale of Shipwreck, Mutiny, and Murder by David Gran, who he also wrote, what is it, Killers of the Flower Moon?
Mhm.
And
Oh,
he
yeah.
wrote the story, The Lost City of Z, which I think I've read. At any rate, this is about an ill-fated, and I think, in my opinion, having read it, ill-conceived British naval military expedition in the mid-18th century that ultimately results after a horrific passage around the Horn in a shipwreck on a desolate island off the coast of Peru, and ultimately what is described as a mutiny. Okay, because the shipwreck, the shipwreck resulted in a sort of a Lord of the Flies type situation where there was significant disagreement about how their situation should be managed. And ultimately, 2 separate groups of individuals made their way back to England and ultimately faced court-martial. Okay, and it's just a gripping story, very well written. The guy is a terrific writer. I devoured this book. So there you go. Good seafaring story.
Cool.
And
I saw
you
this
know,
pick and immediately put it on my list. I've
All
got
right.
to read this.
You'll
This
love
is
it. I thought of
totally
you,
my
Alan. You'll love
jam.
it. Yeah, you will love this.
Alan, what do you have?
I have a game. So this is a video game that just came out at in mid-December. It's called Less is Morse. And if you look into the development of it, you may see familiar name there. So I like playing around with radios, and I know Morse code, and one of the things I like to do is improve my ability at it. But a lot of the available apps and tools for that are aimed at learning the code in the first place. And a lot of the games that are available, or quasi-games that are available, really aren't all that good, or they don't run very well anymore because they're kind of old. So I made a game. And this will teach you Morse code if you don't already know it, or if you do already know it,
Cool.
you can do various, it'll give you the 100 most common words and send them in Morse code, and you type them in and just hit the space bar, and it'll tell you if you got it right. It's a sort of minimalist control panel look to it. I find it fun to play, so I consider it successful on that basis. If other people do, then great. There's a browser version which will probably run in most browsers, may or may not run perfectly. And there are also downloadable versions for Mac, Linux, and PC.
Cool.
So take your pick. And I know that the Linux and Mac versions work fine. And I am told by people who've downloaded the Windows version that it also works. So enjoy, and these are all free.
Cool.
Great.
Cool.
Neat.
My pick is a New York Times article by Apurva Mandavili, and I'm sorry I'm picking New York Times articles. I know they're behind
They're
a paywall.
Yeah.
But I'll tell you what this is about. It's called Citing Misinformation. Florida health officials calls for a halt to COVID vaccines. So the Surgeon General of Florida, Dr. Joseph Ladapo, has said these vaccines are not appropriate for use in human beings. He says the mRNA vaccines are contaminated with small pieces of DNA. And so this article does a very nice job of saying why he is absolutely incorrect to say this, and quotes several people. John Werry says his latest contention is very irresponsible. He has, however, demonstrated a tenuous grasp of science and medicine in general over the course of the pandemic. And who is it at Cornell? John Moore says, you will see a mighty lot of mites in there. There's no evidence that any of this claim of mites actually happens or could ever happen. So basically, there's no basis for him saying that these are not suitable in humans. Obviously, this is some kind of political stunt, and really do not use public health for politics, folks. It's a good article. I like it.
I saw this, and you just want to die. It's just
people will.
Awful. People will.
They will. That's right.
Oh,
this might be the same story that someone wrote me about, and then I reached out and someone sent me a Debunk the Funk episode.
Yeah, he did it.
There's also a Scientific American article debunking all of it with a lot of quotes from Paul Offit, who said something funny like, you're more likely to get struck by lightning than have this, that, or the other thing happen. I don't remember.
Is this an elected official or an appointed official?
He's an appointed official, but appointed by?
Yeah, by.
Anybody?
Ron DeSantis.
Ron DeSantis. Yes.
Yeah, indeed.
Yeah.
Wow.
We have 2 listener picks on Marisa. Hi, everyone. I haven't been listening to all your episodes, so not sure if someone has already shared this as a pick, but if not, I couldn't think of a better group of people to share this with. This is a wonderful animation.
Yes.
Of showing the scale of things from big to down to viruses. It's really well done. I love it.
From human hair to tardigrade to poliovirus. Yeah.
It's good.
It's really well done.
And also we have a submission from Anonymous of a YouTube video. Wow, human cells vibrate with resonant frequency, and it's technically audible. Okay. It's a 10-minute video. You can check it out.
Cool.
That will do it for TWiV 1079. You can find the show notes at microbe.tv/TWiV. Send your questions, comments, picks to TWiV@microbe.tv. If you enjoy our work, please consider supporting us, microbe.tv/contribute. Dixon Despommiers at Racaniello.org, thelivingriver.org. Thank you, Dixon.
You're welcome, Vincent, and everybody else. Great to see you all back. the family is in the nest once again.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
How much snow do you have there?
I don't know. Quite tell. I had to close the blinds. For
Like when you got there wasn't a lot on the ground, right?
There was none. There was none. It was
Okay.
starting to stick on the grass, but it's
Okay.
above freezing, but it's starting to stick.
Alan Dove, alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time. Great to see everybody.
Brianne Barker's at Drew University, BioProf Barker on your favorite social media platform. Thank you, Brianne.
Thanks. I learned a lot, and not just about travertine.
Where do you hang out on Mastodon or
I'm
or
doing
Blue
more
Sky?
on Blue Sky.
Blue Sky. Okay. Same handle, right?
Same. I use the same handle on all of social media.
Okay. I got everybody. I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.